time for questions after.
Okay, yeah, that's what I thought.
So from now on, we're on YouTube, so everything you say is going to be safe. I mean, just so you're clear about this, if there's something about the recording you don't like, we can always edit it later. So if there's anything you say that, oh, that was a mistake, it's not a problem. Obviously, live is live, and we're expecting a few people in the room. You don't have to worry about questions in the sense that I'm going to allow people to join the room and be able to do questions themselves. If there's any question in the chat box, you don't have to worry about that. I'll ask the question if it's pertinent to the point, and if it's not, I'll wait until the end and ask you the questions anyway.
Okay.
Great. Anyway, it's lovely to have you here, and sorry it's not in person. We will definitely have you in person once the world goes back to normal.
Yes, we were just talking that, yeah, hopefully things go back to normal in, sometimes soon.
Yes,
and
this year we hope.
Yeah.
Okay, well, I think I should start bringing people in, David.
Yes.
Okay, here we go.
We're going to wait maybe a minute or two for people to actually settle in. So that we can, start with a full room, virtual room, that is. You want to share your presentation slides, though?
Yes.
So just make sure that you show the whole screen.
Do you see the slides?
I see the slides. Move to the next one just to make sure that things work as smooth as we expect, because sometimes you set the first screen and it seems to be working fine. Yeah, we don't want to have any spoilers. Cool. Sandy, do you think it's a good time to start?
I think it is.
Cool. So I'll start now. So it's a pleasure to have Ivana Bozic, who is visiting us virtually from the University of Washington, where she's an assistant professor at the Department of Applied Mathematics. Ivana has a bachelor's and a master's in mathematics from the University of Belgrade. And then she moved on to Boston to do a PhD also in mathematics, in mathematical biology, I guess, with Martin Novak at Harvard. So she got her PhD in 2012. And she has kept on working on these beautiful mathematical models of evolution and colorectal cancer. And I think we're going to be seeing today some of the latest work. I know that you have published recently in PNAS and in Nature Cancer, and your previous work was also in Nature journals. So I know that you have some really cool and exciting research. So Ivana, please take it.
Thank you so much for the great introduction. Yes, so today I'm going to talk about a recent paper that was published in 2020, already last year, about a mathematical model of colorectal cancer initiation. And the idea for this work was to try to quantify and put into a model the knowledge we have about cancer evolution from the famous Volgogram models and similar, and combine them with newly estimated rates that were experimentally measured of mutations in human tissues, especially colorectal tissues, and rates of growth of benign lesions, and kind of try to see if we put all of these things into a model, can we get as an output something that is similar to estimated prevalence of colorectal cancer and the order of mutations on the way to colorectal cancer. So that was kind of our idea. And now I'm gonna, okay, let's see. Okay. So I have a brief biological introduction into what we know about the initiation of colorectal cancer. And this slide is about cancer in general. So cancer is, we can think of it as loss of normal growth control. which is working really well in normal tissues, in healthy tissues, where the rates of cell division and cell death are tightly kept in balance. But this balance is disrupted in cancer. And so in cancer, we have either an excess of cell division or not enough cell death, so that we have this overgrowth of lesions that are either benign or later on malignant. And somatic evolution can lead to cancer. And usually in most solid tumors in adults, this is a multi-step process where a healthy cell will get the first mutation, which we call a driver mutation, which can cause it to start growing abnormally. It is still not malignant, but it's on its way. And then this population of cells that has a single driver mutation could obtain another driver mutation, and so on. And in the end, this could lead to the appearance of an invasive cancer. And at the end of this process is kind of the most deadly phase, which is metastasis, where cells detach from the primary tumor and travel to distant sites to seed metastases. And so we are interested in modeling the process kind of from this healthy cell to the first malignant cell. And we want to model this accumulation of driver mutations. But in order to do that somewhat precisely, we need to keep in mind that there are two types of driver mutations, so-called oncogenes and tumor suppressor genes. And when we model accumulation of mutations in these two types of driver genes, we have to model them in a different way. And so a simple way of thinking about this is that oncogenes need to be activated in order to become driver genes and promote cancer. And this activation happens by a mutation in a single copy of that gene, but that mutation has to be really at some specific places to promote this oncogenic activity. On the other hand, tumor suppressor genes, their role is to prevent us from getting cancer. So in order for tumor suppressor genes to become driver genes, what actually needs to happen is that they need to be inactivated. So, and that happens if both of the copies typically of that tumor suppressor gene are inactivated. And this can happen by mutations or by something that we call loss of heterozygosity, where a copy is lost, for example. And so these two types of genes become driver genes in very different ways. And so in order to model this malignant progression, we wanted to understand how many driver genes are needed for, in particular, colorectal cancer. And this data here can kind of give us some insight into that. So this is from Vogelstein's Cancer Genome Landscape Review from 2013. And here on the y-axis, this plot shows the fraction of tumors that have a particular number of specific driver gene mutations. So either oncogene mutations, which are in red, or both oncogene and tumor suppressor gene mutations, which are in blue. And so what we see in colorectal cancer is that in most sequenced colorectal cancers, we can find typically a single oncogenic mutation and typically 3 total driver mutations. But especially when it comes to kind of the total number of both oncogenes and tumor suppressor genes, we see that there is a distribution of the number of total driver genes, which kind of has, a median around 3, but can vary. And there are some cancers where there could be up to 8 driver genes. However, there is also this recent paper by Christian Tomasetti and again Bert Vogelstein, in which they look at sequencing data from colorectal cancers in I think smokers and non-smokers, and they are looking at increased rates of and they're looking basically at colorectal cancers that have increased rates of mutations versus those that don't, and argue based on incidence rates in these two types of cancers that actually only 3 driver genes are required for the development of colorectal cancers. And so this is kind of what we are going to go forward, because the 3 driver mutations were kind of typical in that data that I showed you in the previous slide. And with this argument that 3 may be sufficient, we are going to now focus on a model in which 3 driver genes are sufficient for a malignant colorectal cancer cell. And not only that we're going to focus on 3 drivers, we're going to focus on 3 specific driver mutations. And those mutations are kind of the most famous mutations that are that have been known since the 80s or 90s, and which were discussed in the Vogelgram. And these are APC, KRAS, and TP53 mutation. And so it is typically thought that the first step on towards colorectal cancer is a mutation or inactivation of the APC gene. And in fact, APC is mutated in more than 80% of colorectal cancers. KRAS is an oncogene that is also very often in more than 50% of patients mutated in colorectal cancer, and has been typically thought to occur as the second step in colorectal cancer. So we cannot be completely sure about this order of mutations, but kind of from there could be some inferences from experimental data, and this seems to be the most likely order of mutations. And then a later step is this mutation in TP53, which is a tumor suppressor gene, and that which could lead then to carcinoma. So we want to model these 3 mutations, which are the 3 most commonly mutated genes in colorectal cancer. And so now that we have introduced some of the biology of colorectal cancer initiation, we can start formulating our model. And the model is going to be a little complicated, so please bear with me while I try to explain the details of it. So as I said, we will be modeling the accumulation of 3 driver genes: APC, TP53, and KRAS, which are the 3 most common drivers of colorectal cancer. Each of them is present in more than 50% of patients, but together they are present in 15% of colorectal cancer patients. So kind of this model is aiming to explain 15% of colorectal cancer incidence. And other driver genes may be responsible for the remaining 85% of colorectal cancer incidence. And as I said, APC and P53 or TP53 are tumor suppressor genes, and KRAS is an oncogene. And so this on the right-hand side is a schematic of our model. We start from this lower left corner, which represents a healthy cell. We denote this by, for example, TP53++, which means that both copies are active or are working properly of this tumor suppressor gene. And for KRAS, we denote this by KRAS-, which means that this oncogene has not been activated. And then we allow any order of mutations on the way to colorectal cancer. So we do not specify that APC has to happen first and then KRAS and so on. We allow any possible order and then want to see what happens in the model and what the most likely ordering is going to be. So there are many ways to go from this healthy cell to this malignant state, which is here in red, which has both copies of APC and P53 inactivated and KRAS, which is activated. So because we need 2 steps or 2 events to inactivate a tumor suppressor gene and 1 mutation to activate an oncogene, we have 5 steps on the way to colorectal cancer from a healthy cell to a malignant cell. But there are many different pathways in which one can actually do these 5 steps and reach the malignant state. And kind of that is the interesting thing in this model. And yes, I also want to mention, and this is very important, that this work was a collaboration with my postdoc, Shay Patterson, my former postdoc, and Hans Clevers, who is at Hubrecht Institute in Utrecht. So in the previous slide, I've shown you the schematic of the model, that we have kind of these 5 steps and this cube of different paths in order to reach the final malignant state. But as I said already, the model is a little complicated, and it's actually a little more complicated than what the cube is showing. And the complication comes from the fact that there are multiple ways to inactivate a tumor suppressor gene, and we want to keep track of all of them. So these multiple ways of inactivating a tumor suppressor gene are shown here on the right. So if we start from a state that has 2 healthy copies of APC gene, we denote that state with 0 in this first position. And then the first copy of the APC gene can be lost by either mutation or by loss of heterozygosity, which means that we kind of lose that copy completely of the gene. And then once we have a mutation in the first copy, then the second copy could be lost with another mutation, or with, again, this loss of heterozygosity. And these things happen, we assume that our APC is the rate at which a driver mutation in APC occurs. And our LOH is the rate of loss of heterozygosity. And then these rates are halved for subsequent steps because we only have a single copy on which we want to get this second inactivating event. And another thing here that is important is that we do not allow 2 losses of heterozygosity. We do not allow the loss of both copies of APC gene because that's typically lethal for the cell. And so these numbers from 0 to 4 denote different states, different possible states of the APC gene, with the states 3 and 4, meaning that the gene has been inactivated. In the state 3, it has been inactivated by 1 mutation and 1 loss of heterozygosity, and in the state 4, it has been inactivated with 2 mutations. Luckily, the situation is much simpler for oncogenes. So for KRAS, we only have 2 states, which are denoted here, 0 meaning the healthy inactivated state, and then 1 is the activated state, which has this mutation in a single copy of the KRAS gene. And this mutation happens at rate rKRAS. And so I haven't mentioned what happens for TP53, but the situation is basically the same as for APC. only the rates here are different. So TP53 is a tumor suppressor, so it has this complicated way of inactivation. And again, 5 possible states.
Excuse me, when does the aneuploidy kicks in colorectal cancers, and how differences from diploid genome would impact this model's assumptions and outcomes?
That's a very good question, because colorectal cancers often are chromosomally unstable. They have this SYN phenotype. And early works argued that chromosomal instability is a very early event in colorectal cancer. However, more recent studies actually show that typically one needs to, that the TP53 is the crucial event that leads to this widespread chromosomal instability, and that oftentimes you need to have both APC and TP53 inactivated for large-scale chromosomal instability. So we are assuming that large-scale instability occurs after malignancy, after the first malignant cell, and after TP53 is inactivated.
Thank you.
And so we are lucky, and I'm kind of gonna, this is kind of what happens later on, but TP53 is something that happens late, both experimentally, what has been inferred, and also in our model. We're gonna show that this is kind of the latest event. So we can kind of safely assume that we don't have to worry about large scale chromosomal instability in this model. But that's a very kind of good point. So in a sense, we have this model that goes from this healthy genotype 000, and then goes into 5 steps, which could happen in different ways until we reach a malignant genotype, where both tumor suppressor genes are inactivated and the oncogene is activated. But in this model, we actually have 4 malignant genotypes because the way in which tumor suppressor genes can be inactivated can be, as I said, either 2 mutations, which would be 4, let me see here, 4, or by a mutation and loss of heterozygosity, which is kind of this genotype 3. So we have these 4 different malignant genotypes. So now we have specified kind of these transitions between all possible genotypic states in our model. And our next goal was to look in the literature and find experimentally measured rates of mutation, of these driver gene mutations, and of and to see if there's some growth rates also that have been measured for these premalignant lesions. And so in order to calculate a specific driver mutation rate, we look into each driver gene separately and try to calculate the number of positions in which that gene can be mutated so that it will produce a driver mutation. And this is again a plot from Vogelstein's review from 2013, in which you can see the large difference in the patterns of mutation in oncogenes and tumor suppressor genes. So this PIK3CA and IDH1 are oncogenes. And these red triangles are showing kind of where mutations in cancer patients happen in these genes. And you can see, particularly in this case, in the case of IDH1, that there is a specific site that typically gets mutated for this to become an oncogene. In this other oncogene, there are a few more sites, but again, there's maybe 5 specific or 4 specific sites where the gene really needs to be mutated in order for this activating mutation to occur. On the other hand, RB1 and VHL are tumor suppressor genes. And basically, there's many more ways in which one can inactivate a tumor suppressor gene. Basically, you want your mutation to produce a stop codon somewhere in the gene, and that will inactivate a copy. And you can see that the distribution of mutations in cancer patients in these two tumor suppressor genes is basically all over the gene. And so we are actually able to calculate for both APC and TP53, based on their genetic sequence, the number of positions that, if mutated, would lead to a stop codon. And we use that calculation in order to calculate the number of driver positions in these tumor suppressor genes. And then for the KRAS oncogene, we look in the literature to find the estimated number of these specific hotspot positions that, if mutated, would lead to this driver activity. So, but in order to, so we use the following formula here for the driver mutation rate. RG, which is the rate of mutation of the driver gene G, is the number of positions, of driver positions in that gene that I talked about in the previous slide, multiplied by the point mutation rate, U. And so our next goal was to find an estimate for the driver, for the mutation rate U. And we actually learned about this estimate from Hans Clevers, who was the author on this paper, in which they took colorectal, intestinal, and liver stem cells and expanded them into organoids and then sequenced them. And what this was able to do was, because they looked at patients across different ages, they were able to get an estimate for the rate of accumulation of mutations during human lifetime. And what was striking was that across these three different tissues, they found a very consistent rate of accumulation, which was 30 mutations per year per whole genome. And what we do is we just calculate what is the corresponding point mutation rate in the exome, and then basically use that as U, which will help us get these driver mutation rates. And so this is normal mutation rate in healthy tissues. And so our assumption is that during the pre-malignant tumor evolution, the mutation rate remains more or less the same as the healthy mutation rate. And that if there's a large increase in mutation rate during cancer evolution, it happens after this malignant transformation.
Ivana, before you go on, can I ask a quick question?
Mm-hmm.
Why is the age distribution here so strange?
That's their donor. They didn't have that many donors. And so I think that is what leads to this strange distribution.
Right, okay. So the fact that there's some young and some old in colon, then some in between in small intestine, then only in between for liver is just a product of the source of the data, basically.
Exactly.
Okay. And so this is for all mutations?
This is for all mutations in the genome.
Right, okay, thanks.
And so basically, we have now been able to describe the mutation rates from the literature and to have good estimates for mutation rates. Now we want to look into what is the starting healthy population in which this process is going to play out. And kind of one of the key things that is helping us make our model be not too complicated is that we are focusing not on single cells, but we are focusing on colorectal crypts in this model. And the number of healthy crypts has been estimated to be about 10 to the 8. And this is our starting population. We are assuming that if a mutation occurs in a cell inside a crypt, that it will relatively quickly either be lost or it will expand and become clonal in that crypt. And this stabilization time for crypts has been estimated to be on the order of 1 year, which is relatively short compared to the time of this process. So we are basically just looking at how mutations accumulate in crypts. And we think this is justified because also the previous estimate of the mutation rate has been looking at, you basically can measure mutations that became clonal and fixated and stayed in your population. Kind of the mutations that were lost are not accounted for in that mutation rate. And so for those who are not very familiar with crypts, I'll just say a few words that basically the colon consists of these kind of millions of crypts, each of which contains about 1,000 cells. And on the bottom, we have around 5, maybe to 10 stem cells, which when they divide, replenish the crypt. Then we have these transit amplifying and then finally differentiated cells, which cannot divide and kind of just die and are flushed out from the crypt. And so the first thing that we do is we study our model using computer simulations, starting with 10 to the 8 healthy crypts, which have genotype 000 at time 0. And we have 50 different genotypes, so we keep track of all the crypts with all different genotypes and their numbers. But we also track paths that are taken by individual crypts. We are interested in not only when we reach malignancy, but what are the likely paths through which we reach malignancy. And so even though we have 50 genotypes, if we want to keep track of different individual paths, that actually leads to 270 distinct paths to our malignant genotypes that we also keep track of. And we use 2 algorithms to simulate this model. The first is the famous Gillespie algorithm, which gives us basically an exact simulation of the process. The problem with the Gillespie is that it's very slow because each update happens, update for each single crypt happens, one at a time. So it's super slow if you have 100 or 100 million crypts. So we also use a tau leaping algorithm, which is an approximation also by Gillespie of the Gillespie algorithm, where updates happen only at discrete time steps. But we have shown that these two, once you choose your time step wisely in the tau leaping algorithm, that these two are really give very, very similar results.
Hi, Ivana. Quick question, Philip here. Is in the tau leaping, is that an exogenous parameter you set once, or is it somehow adaptive to the state of the population?
It is an exogenous parameter that we set once.
Okay, thank you.
Sure. And so in addition to the simulations, we also try analytic approaches to understand this process. But in order to reach some reasonable analytic results, we actually make some simplifying assumptions. So we simplify our process first in that we neglect the double.
Sorry, before you go on, just to clarify, you maybe already said this. There's no split space considered here, right? There's no crypt architecture.
No space is considered here. And because we are only modeling single crypts, yeah, so that would be probably a 2D space that one would consider.
So basically you are looking at the entire crypt, but you're assuming it's just like a well-mixed thing. Is that right?
So we are assuming our agent is a crypt. So basically, we don't look at the population of cells within the crypt. We're just saying, either a mutation is fixated in the crypt or it will be lost. So kind of our single agent in our model is a crypt.
Right, understood. So that's the lowest resolution you're at, is a crypt.
Exactly.
And then you assume that the crypts are just counted. They're not in, like, there's no crypt fission or anything like that.
There will be crypt fission. I'm going to be getting there, but there's no space.
Right, thanks.
Sure. Yes, and so that's a very good question. So that's something that I'm still going to talk about. So we are neglecting in the analytical approximation double mutation of tumor suppressor genes, because that's much less likely than mutation and loss of heterozygosity, because loss of heterozygosity typically happens at much higher rates than mutations. So this lowers our number of genotypes for tumor suppressor genes from 5 to 4. And now we only have a single malignant genotype, which will be 331. And another approximation that we are doing is we are using a mean field approximation to calculate our main quantity of interest, which is the probability that a cancerous crypt is present at time t. So this is our P of t. And so we have this kind of approximation where we say that in order to calculate P of t, we look at the expected numbers of cells at genotypes that are just next to this malignant genotype. So the pre-malignant genotypes are 231, 131, and so on. And so these numbers like N231 will be the expected number of crypts of that genotype at time t. And then we multiply this by the rate at which you go from that pre-malignant genotype to the malignant genotype. And so this we call this the mean field approximation, and it leads to this differential equation for P of t. Sorry. So kind of in order to calculate this probability, now we need to find these expected numbers of cells of pre-malignant genotypes at time t. And now coming back to Sandy's question, is there splitting of crypts? And the answer is yes, there is. So these pre-malignant genotypes will sometimes lead to abnormal growth. And APC inactivation was shown to lead to increased rates of crypt fission. And that rate has been measured a few years ago in human tissues. And similarly, KRAS activation was also shown to lead to a small increase in the rate of crypt fission. And that was measured, I think, 2 years ago. So we use these two estimates. And we also use the fact that TP53 was shown not to lead to selective advantage on its own. So we will assume that P53 on its own will not lead to any increase in crypt fission. And so how we incorporate this into the model. So we allow crypts that have inactivated APC to divide according to a pure birth process, stochastically, with rate B1. And I didn't say this before, but when I talked about this rates of mutation, we always assume that these mutations happen in a stochastic manner. And also crypts that have activated KRAS are dividing with rate B2. And then if crypts have inactivated both APC and KRAS, we let them divide with rate B1, 2. And so to find the expected numbers of these premalignant crypts, we analytically solve a system of 31 differential equations describing the expected number of crypts with premalignant genotypes. And in order for our solution to be simpler but without loss of accuracy, we neglect negative terms in each equation, the outflow terms, as they don't significantly affect the average dynamics. So for example, for genotype 001, which is wild type for APC and TP53, but mutant for KRAS, we just have this simple equation that basically the change in that population is due to that population dividing with rate B2, and due to this inflow term from mutation in KRAS from the healthy crypts. And we neglect that basically some of these 001 crypts will be lost due to mutation and become other genotypes, because mutation rates are small and we can neglect that without virtually any loss of accuracy. So then we solve this system, we get this analytic results and plug them in as input into this mean field equation for the probability of malignancy. And so what we do is we actually calculate, even though we know that both APC and KRAS have been shown to provide selective advantage and to increase crypt fission, we also try to calculate what would happen in the scenario where all drivers are neutral. And this is our scenario 1. And that means that there is no crypt fission, basically, just transitions between genotypes. And this would be the scenario where these mutations are neutral prior to malignant transformation. And that's a model that some people have argued for as well. Then we study the case where only APC has selective advantage, which means that this B1, which is the rate of division of APC crypts, is positive, and B2, which is the rate of division of KRAS crypts, is 0. And finally, this is our full model, where both APC and KRAS have advantage. And we have these 3 relatively simple equations for the probability of a malignant crypt at time t. So what is interesting is that in the neutral model, basically we have a polynomial in time. And then for this other 2 cases, we have something that is kind of like an exponential. So in this plot, we compare our analytic formulas in the 3 scenarios, which are shown by lines here, to computer simulations, which are shown by these crosses. And so I want to point out that the parameters we are using for the division rates of crypts are 0.2 per year for APC, which was estimated in this Baker et al. Paper in 2014. And for the rate of division of KRAS crypts, we use this 0.07 per year rate, which was estimated recently by Nicholson in 2018. And
One quick question. So the simulations here, they're the tau leaping.
Yes, they're the tau leaping.
Thanks.
We also compare tau leaping to Gillespie and show that they agree.
Yeah, I think you already mentioned that. Yeah, I just,
This is tau leaping, correct? And what we see here is that for this intermediate model where APC only has advantage, our simulations and our analytic calculations agree pretty well, and that in the full model that we are mostly interested in, the analytic calculation is decent up to maybe 65, 70 years of age, after which it starts to overestimate the probability of malignancy from the simulation. And another thing that we show here is that in this red point here, we plot the lifetime risk of colorectal cancer multiplied by 15%, which corresponds to the fact that only 15% of colorectal cancers have all these three mutations. And we compare that lifetime risk with the probability of having develop colorectal cancer by age 80 in our model. And they agree pretty well. And so unfortunately, we cannot compare the entire incidence curve to our model because we do not have information on the or from epidemiological data on what mutations these patients have. So we can only compare lifetime risk at this point. But kind of it is encouraging that the lifetime risk agrees with the lifetime risk from our model. Some other things that we are also able to.
I got a quick question.
Sure.
I guess you're aware of like Peter Campbell work that they actually have the temporal data on colon cancer, even from the same patient, and their numbers are a little bit different from you, but you can simply, I guess they just published it in, I guess, Nature last year. I guess that would be like perfect experimental data that you could actually, not experimental, from patient that you can actually test your model.
Yeah, I'll try to find it. If you don't mind, if you could send me the paper, that would be amazing, but I'll also look for it.
Absolutely.
Thank you. Yes, so as I said, some other things that we could calculate are the expected numbers of mutated crypts and that have these different mutations. And so for single mutations, here we plot using these formula on the right. Here we plot in blue the number of crypts over a lifetime that have fully inactivated APC. And in red, for example, is the crypts that have activated KRAS. And so the interesting thing is that because KRAS only needs a single copy to be activated, initially the number of KRAS mutated crypts is the highest. But because APC mutated crypts have higher growth advantage, around age 40, the number of APC mutated crypts actually becomes higher than the number of KRAS mutated crypts. So even though you need 2 steps to inactivate APC. And this shows kind of the importance of the selective advantage that APC provides in colorectal tissues. Another thing we can also calculate is the number of doubly mutated crypts that have 2 driver genes. And the formulas are here in the bottom, and we plot them on the top as a number of crypts as a function of age. And the highest number of doubly mutated crypts will be those that have driver mutations in APC and KRAS, which are here in purple. And kind of we understand this, that it's a consequence of the fact that these 2 genes provide the largest selective advantage. Whereas these other 2 doubly mutated genotypes are basically, expected numbers are lower than 1 throughout most of the lifetime, and that's because they contain p53, which doesn't provide any selective advantage. And the final result that we were very excited about was about the order of mutations on the way to colorectal cancer. And so, as I said, our simulation contains information about the different paths that were taken by cells that became malignant. And we aggregated those data and looked at what was the most likely order of mutations on the way to colorectal cancer. And what we found is that in over 2/3 of simulations, APC inactivation was the first event, followed by KRAS mutation, with TP53 inactivation being the last event. And so we were very excited about this because even though we didn't assume any order in our model, we actually are able to obtain the most likely order of mutations that people think, and that has experimental support for what happens in colorectal cancer. So that was very exciting. And so we also wanted to understand analytically the order of mutations, and again looked into our mean field approximation because we wanted to understand what is causing this mutation order and what are the important parameters that determine mutation order. And so we looked into, again, this mean field approach, and we show that it's sufficient, even though we have this complicated cube of genotypes, that if you are interested in the probability that a certain path is taken, because the outflow terms are very small and they can be neglected, you can actually just look into rates on a single path in order to calculate the probability to reach this malignant state using a particular path. So to calculate the likelihood of a path, so we look into this model that starts from a healthy state, which is state 0, and then in 5 steps can reach the malignant state, which is here in red. And then we assume, because we want a general result, that the rates of mutation that you can go from one to the next genotype are given by these R's. And then we assume that there are some division rates of each pre-malignant genotype, which we denote by these B's. And some of them can be 0, and some of them can be greater than 0. And so what we showed that for a path like this, the Laplace transform of the probability of reaching that malignant state through this path by time t is given by this formula. And so we are using Laplace transform because some of these B's can be 0, and this makes our calculations much easier. And what we have in the formula is N0, which is the initial number of crypts that are healthy. Then we have these mutation rates that are all multiplied together. And then in the denominator, we have something that depends on the division rates. And so because in our model, this product of mutation rates and number of healthy crypts is constant, is the same for every path. The probability of path is maximized when you maximize what is this here, which only depends on the division rates. So basically, you maximize the likelihood of that path, and that that maximum value does not depend on mutation rates. It only depends on division rates, on the growth rates. And in our model, this happens when the growth rates on that path are maximized. So that means the most likely path is the one that collects mutations so that your fitness is always the highest possible. And that happens when the order of events is inactivation of one copy of APC, then of the second copy of APC. And this being first means that you suddenly have this greatest growth advantage that is provided by APC. Then you collect KRAS, which again improves your growth advantage, and then TPP3, which will not help you grow faster before, but which may be very important for malignant transformation and kind of the later evolution. And so kind of something that you could see also from the previous slide is that if we look at what is the most likely path and what is the likelihood of a certain path to reach malignancy, in our model, it actually does not depend on mutation rates. And the reason is that the probability of each path is proportional to this product of mutation rates and initial number of healthy crypts. And that product is the same for every path in our model. And so the probability that a single path X is taken, conditional on that we reach this malignant state, is basically when you divide the probability of that path by the probability of all other paths. And so in this expression here, each of these probabilities has something like this, a product like this, which cancel out. And so in the end, the likelihood of a single path is basically independent of mutation rates in our analytic approximation. So that was pretty cool. So kind of that seems to imply that the mutation rates are really much less important in this process when it comes to the ordering of driver events than the growth advantages that they provide. So this would kind of we conclude that basically the order of driver mutations is the order in which you have the maximum fitness possible.
Sorry, got a quick question. Going back to Sandy's question, actually. So you keep saying crypt, but the rate that you're showing is the rate of mutation in single cell. And you said that your crypt is not a single cell. It's can you clarify on that a little bit, please?
Yes. So the rate that we use is the rate of accumulation of mutations in stem cells. Which
a crypt can have like hundreds of them or 10 of them.
No, it will have a few, but basically it's the rate at which successful mutations accumulate in crypts. So in stem cells. So
only stem cells in the crypt, not any other cells, just the stem cells in the crypt.
Yes.
That carry this mutation.
Yes, but basically the thing is that the rate that has been measured in that paper that I showed you measures only mutations that fixated, that remained in that stem cell. So basically only it's the rate of successful mutations in stem cells. And that's what we are interested in, because once a mutation is present in a single stem cell of a crypt, and say you have 5 stem cells, that mutation will, within a few months, typically be either lost or it will expand and be present in all stem cells and then in the entire crypt. And so in a sense, we are neglecting that fact that, some mutations can be lost, some mutations can fixate, and we're just modeling the mutations that will fixate in an entire crypt.
So it's basically the effective mutation rate in the crypt.
Correct. Exactly.
And do you have different number of stem cells in each crypt in your model, or is it like?
No, so for us, we are not modeling what happens in the crypt. So lowest resolution, as Sandy said, in our model is a single crypt.
Which contains how many stem cells?
So in reality, it can contain
No, no, in
5
your model.
stem cells.
In your
But
model.
in our model, we don't look at what is contained in crypts. We just say a single crypt is what we care about.
Thanks.
And so the final part of the model is, or of the results is that we just wanted
keep pushing on that point. It's not actually known, right, how many stem cells there are.
That's true. I mean, people have been estimating them, and I think 5-ish to 10 is the most likely, but nobody knows exactly.
Yeah, thanks.
Yes, so we just wanted to compare to check in simulations whether probability of a certain path to malignancy does not depend on mutation rates. And so we found that this is true in our analytic approximation, but we wanted to make sure that it still holds in the simulation. And so we look into 4 different scenarios. Scenario one is this scenario that has the rates, the parameters the same as in our model. In scenario 2, we double all rates, all mutation rates. In scenario 3, we halve all mutation rates. And in scenario 4, we keep all rates the same, but multiply one rate by 10 to kind of make it be asymmetrical. And here we show the relative probability of different paths to malignancy. And what kind of the takeaway from this plot should be is that even if the rates are very different in these 4 scenarios, kind of the likelihoods of different paths, which are shown here, are pretty similar. So in all 4 scenarios, we have these 4 most likely paths, which have similar relative probabilities, and these differences could be due to stochasticity and us not running enough simulations, because especially when rates are very small, things become very small to run. It's very, very slow to run. But kind of the idea here is that we were able to confirm, even in simulations, that the mutation rates do not have a significant effect on the order of mutations on the way to malignancy, but that basically the most important thing are these growth advantages. And so that was all that I wanted to say today. This is a summary that we developed this as much as possible biologically realistic model, a stochastic model of colorectal cancer initiation. We find that the reported lifetime risk of colorectal cancer can be recovered using our model and experimentally measured parameters. And we demonstrate that the order of drivers is determined primarily by the fitness effects that they provide and not their mutation rates. So thank you all so much for listening.
Thank you very much, Ivana. This was a great presentation. It's a great example of how hypothesis-driven simple models can actually make great contributions in cancer research. I heard lots of people asking questions during your talk, so I don't know if there's a lot left, but I know that Mehdi or Andre might still have some questions. I know that because Mehdi's still talking on the chat. Mehdi, do you have any more questions?
I'm trying to understand. I mean, Sand is helping me because I don't want to, like, exhaust the audience. I mean, because I know the work of Peter Campbell pretty well, and they're looking at the mutation rate in, like, the longitudinal data from the patients, right? They even started from, like, the egg, like astronaut, the zygote. Astronauts, like, this one single cell star, like they sequence that, and then they have like mutation rate at time 0 versus like 5 years, 10 years, and they show that some of the simple inflammation can actually increase some clones, or because the crypt can actually um, duplicate themselves or increase themselves. So like the mutation rate can be also like affected, and the clonality, subclonality. So that's why I was a little like, confused about like, one crypt, having, being a representative of like a stem cells in there, and the mutation rate of in all these somatic cells, I basically is the same as they showed in their work. So, but I think I just need to read her work a little bit more.
Okay, so it wasn't as much a question as an expression of interest. Okay.
Yeah, that was, I just, said my thoughts like out loud. So I wasn't really sure. Like I was trying to understand the whole thing.
But I also wanted to mention that Magdi mentions an important point, which is inflammation. And that's really important in colorectal cancer and can lead to things changing. And this is something that we may want to include in subsequent work. It has been shown that if you have inflammation, TP53 mutation can actually lead to fitness advantage. And so also, if you have some familial syndromes that predispose to colorectal cancer, that can also change the model and some rates. And so kind of this is the simplest scenario, but one can add layers of complexity onto that. And this is something that we're thinking about.
Thanks, Ivana, because actually that links the two questions I had for you. The first was your thoughts on the macroenvironment, and the second was what your future work was going to be. So that kind of answers both. So what about aging?
That's a very good question as well. And I have some conflicting thoughts about that. So I know like James de Gregory's work showing that, aging increases incidence of cancer. But then on the other hand, there is this work by Tomasetti and Vogelstein that says that mutation rates actually slow down with age. So
yes,
I have some confusion about the effect of age.
It could be that both things are right, but which one is more prevalent and more important in particular context?
Yes, but that's also another thing that could be added to this model.
Cool. So
I want to maybe quickly, Philip again, dig deeper into this one comment about exactly kind of add additional modes of environment, especially say inflammation. Have you begun thinking about this? How well-defined genomic aberrations as you or genetic aberrations as you track them in concert with, say, immune surveillance could contribute to additional accuracy, say, and by what amount to this kind of to the risk and over lifetime?
Yeah, no, that would be amazing to do, and as you.
But where to start, right?
Yeah, and also
where and how.
In this model, I tried to include things for which we had experimentally measured parameters. And so kind of the goal for the future is to try to find more such things. Mm-hmm. But as we try to do more complicated things like inflammation, immune system, and so on, having experimentally measured parameters becomes more tricky to find. So I think that will be the difficulty. Like, I think setting up a model will not be so tricky, but kind of actually having it be realistic and with some kind of experimental backup will be the tricky part.
Maybe the model could inform how to look for it.
Of course.
That's
if you just put in some more fundamental, and I mean, if you just go back to databases, you could correlate, say, those drivers to other markers of immune surveillance and escape, and come to some preliminary conclusions of likelihood of seeing those paths together with the other events, and then come up with some preliminary conclusions, and that could help set up a more refined study.
Yeah, that's a very good point.
So something else that I think like about this question of having like a single mutation rate for a crypt altogether. I think if you're assuming that there's no selective advantage given to a particular stem cell when it acquires one of these new driver mutations, then that assumption is fine. However, if one of these drivers allows or gives a higher likelihood that a stem cell will outcompete the other stem cells in the crypt, then it might make sense to like artificially increase that mutation rate to account for that. If any one of these stem cells gets that mutation, it's likely that it will take over. I'm just wondering if you've thought about adding something along those lines. It might be hard to get actual data about that, but. That's
a very good point, and we actually do that. But I kind of, for simplicity, I didn't mention it, but we actually use, and that's from mouse data, but it has been measured that if an APC mutation appears in a crypt, what is the probability that it will take over? And so it will be higher than by chance. And we multiply our mutation rate for APC by that increase in the chance of fixation. And we do that also for KRAS. So we kind of try to account for that as well.
Perfect.
Won't those rates change temporally as you get feedback with the microenvironment and you have an underlying shift in the stem cells though? So as cancer progresses or is initiating, you tend to have a growth in the pool of stem cells, like AFAP, FAP patients, like you were talking about some of the inflammation, HMPS, some of those diseases that cause larger numbers of stem cells at the base of the crypt. It'd be interesting to look at how, if you have a temporal adjustment to those rates, how it would change these findings. I don't know.
Yeah, that's something that we didn't look at, the potential increase in the stem cell pool, or as you said, temporal changes in the microenvironment. So kind of thinking about how to include those, some things like those in a smart way is kind of what we're thinking about.
Cool. Well, thank you very much, Ivana.
Thank you so much for the invitation. It was so nice to see many familiar faces after a long time.
Right.
Hi, Philip.
And hopefully next time we'll be here in Tampa.
That would be amazing.
And we can all go to Disney World.
Yay!
No annual passes anymore, Philip.
No.
I prefer the beach, but Disney World is fine.
Yes. No, there are better beaches even than, yeah.
I'd rather go to the beach as well. Well, see you all guys. Bye.
Thank you very much.
Thanks. Bye.